Vamos con la siguiente presentación. Para mí es un gusto y un honor muy grande tener aquí a tan distinguida persona como es John Butler. Para aquellas personas que han estudiado o están en el mundo de la genética forense, yo digo que obligatoriamente tienen que haber leído alguno de sus libros. Y si no lo han hecho, están perdiendo el tiempo. No me voy a detener mucho en su currículum, que es muy vasto, porque John tiene numerosas publicaciones, ya lo había mencionado también, que existen también varios libros. Y John en particular, bueno, te agradezco enormemente la aceptación de nuestra invitación. Él actualmente se encuentra como asistente adjunto al director de ciencias forenses del grupo NIST, que es muy importante para la genética forense. Y bueno, pues yo ya dejo de hablar para que hable la persona que realmente es importante que lo haga. Gracias, John. Thank you, John. So, go ahead.
Okay, thank you very much for the invitation to speak. I haven't prepared a lot of slides, and hopefully the slides will be shared with everybody, the PDF that I provided. And then if that's helpful to do further interpretation for everybody or translation into Spanish, then I'm sorry, I can't speak Spanish. That's something I'd like to learn better, but I don't have that yet. So to begin with, I would like to say that the presentation is my opinions and don't represent those of where I work at NIST or other places where I've spoken. And then also anything when I identify a specific commercial entity or a commercial something commercial, I'm not endorsing that. That's not something that NIST does, commercial products. So I'd like to speak a little bit about background and available resources. Just a little bit in terms of background, I'm from the National Institute of Standards and Technology, and we are part of the U.S. Department of Commerce, and we are trying to help improve industrial competitiveness. And our core values are around perseverance, around integrity, inclusivity, and excellence. So making sure we try to get the best results from things. We are located near Washington, D.C., in Gaithersburg, Maryland, and we have more than 6,000 employees here and supply lots of reference materials. And we also define the official time for the United States as part of our role. Our director is Lori Locascio, and we also do a lot of work to try to help improve computer chips efforts. So in the forensic science program at NIST, you can go to the website to see more. We have in the special programs efforts in research, in standards, and in foundation studies. So in research, we cover areas like DNA, forensic genetics. And the Organization of Scientific Area Committees is working on standards, and these are in the various areas of forensic science. And lots of people participate in this. That's led by someone named John Paul Jones. And then I'm in charge of foundation studies, which is where we're looking at understanding the background of each of the disciplines in forensic science. So DNA mixture interpretation, which I'll talk about today, is something we've studied quite a bit. We also have done work in digital evidence and in bite mark analysis and working on firearms as well. A little bit about me. I work in the Special Programs Office at NIST, the National Institute of Standards and Technology. I have 6 children, and I work on research and write articles, and then review other people's work, and then teach, explain things to people like I'm doing now. I worked at the FBI laboratory for several years and the Armed Forces DNA lab, and I've been at NIST for most of my career. Some of you may know me from the website, Starbase, the short tandem repeat DNA database. I made that about 25 years ago. I've also written a number of textbooks on DNA that some of you may be familiar with, and there's a YouTube video of me talking about the process of writing. And most recently, I wrote a book called Understanding Forensic DNA. And in that book, we talk about, my co-author and I, talk about a DNA interpretation of mixtures and the curse of sensitivity. And one of the challenges that exists today is because we're so sensitive with DNA testing that it makes it more difficult to interpret the evidence from that. So that's described in this book. So if you're interested in learning more about this, I point you to reading the details in that book. There's also something that the International Society for Forensic Genetics has been working on, is making resources more available in other languages. So there's a document, if you weren't familiar with this, I encourage you to go click on the links, the Sense About Science website, and it has a lot of details about forensic genetics that are available in Spanish, so you can learn that make sense of forensic genetics that way. And I invite you to join ISFG. I am currently the president. We do have a Spanish-Portuguese speaking working group, and we have people receive the journal of forensic genetics. You had a chance to come to meetings. I dressed up as the president, George Washington, when they came to Washington, D.C. 2 years ago. And our next meeting is going to be in Spain. So I encourage you, if you're interested in learning more about ISFG, come to the meeting and come in Spain to learn more, and you'll be able to talk to Spanish colleagues there in Spanish to continue to learn more about the field. There'll be a proceedings that comes out of that conference as well that will be available for people to learn from. But the talks will be in English for that. In terms of mixtures, we have prepared a nice resource on that's available on the NIST website that talks about DNA mixtures. And this covers topics like why are mixtures so prevalent or occurring so often in cases, in forensic cases? Why are they difficult to interpret? What are some of the uncertainties that exist there? And how can software help in many cases? And this, if you do a Google search for DNA mixtures, this is one of the top things that you'll find. And this, we've also been working on process maps where we've looked at, this is something that was done 2 years ago. It's a process map that looks at each of the decisions that are being made in DNA testing. So this might be a helpful resource if you're starting a laboratory or if you're trying to train people in your laboratory about DNA testing. I just wanted to make you aware of these resources. And this, you might have for example, this is on one of the pages that breaks out the decisions that are being made. Were multiple tests requested? Is this requested test validated? Has it been studied before? And most recently, just last month, there was a big report that came out, a 436-page report. And this is on DNA interpretation in terms of the human factors. And so what this talks about is looking for information on, there's a section on education and training. There's a section on reporting and testimony, and on the research culture and the research needs and so on that are there. So I refer you to this resource as something that would be very useful to learn more about what's going on in terms of the discussions and some of the things that are happening, the questions that people are asking when they look at DNA testing. And then the draft report that we published 3 years ago, we're in the process of finalizing that. And there is 250 pages of information on DNA mixtures there. And I'm going to talk about some of those pieces today in my presentation. And we collected a number of public comments from people over the 6 months following the report being released. And we'll release a final version when these revisions are completed. So a number of things have happened in the last 3 years since our draft report came out, and particularly related to Chapter 4, which talks about reliability issues. There's more details in some of the papers that are appearing in terms of probabilistic genotyping. And one of the examples is this paper listed here from Adamovich in 2022 that looks at a new software called Master. And then another thing that's been going on is there's lots of research now and more that's come out on understanding DNA transfer and the persistence of DNA, the prevalence and the recovery of DNA. And these articles provide more details in terms of activity level questions. We'll get into this more in the presentation. And then the report that I just mentioned, the NIST DNA Human Factors report, Chapter 7 talks quite a bit more about this transfer, persistence, prevalence, and recovery, and some of the issues that exist around that. If you compare these reports that we've had, the Human Factors report there on the left covers DNA interpretation. It's a joint effort with not only NIST, but the National Institute of Justice. and it's building on previous human factors work that's been done. This was written by a very diverse committee and then had later input from external reviewers. It was released as a final version that we just saw, a month ago. There was no public comments before finalizing it. It's large. It's 12 chapters covering a large range of issues, over 400 pages in length, and it does include actionable proposals to strengthen the field. On the scientific foundation portion that's there on the right, we have a report that's focused on DNA mixture interpretation. This is a NIST-led effort written by NIST authors with early input from a resource group of external people. We've released a draft, and I put a hyperlink there to the draft that we enabled people to provide public comments to. And that's, in the end, it's going to be shorter than it was. Originally, it was 250 pages. It'll be a lot shorter because we're putting some of the material into the supplemental documents that will go along with it. And again, it also issues actionable proposals for strengthening the field. And what people do, we'll see going forward. But we hope that both of these reports are useful and allow people to better understand how to improve the things in the field. So let's talk briefly about, as we get into mixtures now, is two things that are really important to keep in mind. Is that number one, mixtures are going to be inherent to many forensic samples. That's just the reality of forensic evidence, that you're often going to be facing mixtures. The second point is that there's multiple approaches that have been used for this mixture interpretation over time. And there's even today, laboratories will do that in different ways. So one of the things that has been done with sexual assault samples is we can use a technique called differential extraction that was first described in 1985. And that can help physically separate sperm cells from epithelial cells if you're trying to work with a sexual assault sample. And then the second part of this is we have mixture deconvolution, where using peak height or peak area information, we can separate things out. And this was described in 1998 with two-person mixtures. And there's been a lot of work done with software to help with large, like 3 or 4-person mixtures since that time. In terms of what's been done for approaches for interpretation, we have likelihood ratios that have been considered for mixtures since 1991. We have the combined probability of inclusion that was initially described in 1992, but it only works when there's no alleles that are missing. So you have to have a complete data set, which is unlikely to be occurring when you have low levels of DNA samples. And this results from having high sensitivity systems used today. We're going to detect more DNA. Therefore, we're going to be dealing with situations where there's going to be missing information. And this is why the CPI, or the combined probability of inclusion, does not work in these types of situations. So you have to approach it differently. And finally, the likelihood ratio has been incorporated into probabilistic genotyping software, which I'll sometimes abbreviate as PGS. And these systems have been steadily growing in use in forensic laboratories over the past decade. So they've been introduced pretty widely in the last 10 years. Now, in some places in the United States, this is the primary way that testing is being done for mixtures. Okay, so if we give it just a brief history of mixture interpretation, we have the measurement aspects. We originally began in the field with single locus probes for doing testing. And this is where you'd be testing different sections of the DNA with restriction fragment link polymorphisms. Then there was early work done with PCR, and the polymerase chain reaction is a way of copying the DNA. That was originally done with a polymarker, or PM, and DQ alpha. And these were systems that were not they didn't have lots of possibilities, and therefore they didn't work as well on mixtures. And then for the last more than 20 years, we've been using fluorescently labeled short tandem repeats, or STRs, and these enable you to look at lots of different alleles at many markers. So we have the variable number of tandem repeats that were done with RFLP. Here we have poor sensitivity because we didn't have PCR. There's lots of alleles, but there's only a few markers or a few loci that are being tested. We did single nucleotide polymorphisms, or SNPs. Here we had improved sensitivity, but when they were originally being used back in the early 1990s, there was few alleles and few loci. And then finally, we've been using short tandem repeats for the last several decades now. This is high sensitivity because we're using the PCR process to copy the DNA. We have many alleles, sometimes with more than 12 alleles at a locus, many loci. Many times we're doing 20 markers or 20 loci at a time. And so this has become the primary method that's being used today. And then we have where people are moving to now is next generation sequencing, or sometimes called massively parallel sequencing, or MPS. And this is where we have, again, high sensitivity because we're doing PCR. There's more alleles, more loci. You can do both STRs and SNPs and microhaplotypes. So it gives you more capabilities. It's more expensive, and depending on where you're testing, you may not be doing it as much. There's a number of approaches I've mentioned. Like earlier, we have the CPI has been the common approach in the US for many years. More people are moving now to likelihood ratios and to probabilistic genotyping systems. And then there's been work done for many years with random RMP, or the random match probability. And that's been done for deconvoluting major components of mixtures. And then finally, we have just mentioning here, the software. The theory was first described back in 2000, but it's only been really introduced in a large measure in the last 10 years as software programs became available. And these are some of the cases that have been important for the growing use of DNA. The O.J. Simpson case really emphasized the ability of collecting your samples well. The Oman bombing case, which happened in Northern Ireland, was focused around the DNA transfer capabilities and also low levels of DNA. You had the Amanda Knox case, which was surrounding the issues with DNA transfer, and then the Gissentanner case, most recently, is one focused on around probabilistic genotyping software. So as we look to where's the future going in this area, again, I'm just giving a high-level overview, but there's a lot of work going on now to try to pick cells apart. And so instead of looking at all the cells together that might come in the sample, can you pick them apart and then get the individual components? And so these are just some of the papers that have been published in the last 2 years on this topic, where people are looking at trying to do single cell work. And so we'll see how that comes into play into future forensic laboratory work. But I just wanted you to be aware that there's new efforts in this area of looking at single cell analysis. And in chapter 6 of our report, we'll describe the sequencing and the microhaplotypes and stuff in more detail. Okay, so I want to turn attention. So that's kind of giving you background information, both the history as well as other resources you can go to learn more. I want to focus our attention now on kind of key concepts that are important when you're looking at doing DNA mixture interpretation. So in this case, I'm going to focus on some of the things that we learned as putting together the foundation study, and also the ISFG DNA Commission article was published in 2006. I really look at that article, which is published now almost 20 years ago, as being a core foundation to look to for DNA interpretation. And in fact, all the things that were discussed in that report are still applied today. So let's go back to this is not the final version, but this is the way that we worded it in the draft report for our foundation study. That the important principle here is that not all DNA samples are equivalent. So the key takeaway that we had with this was that DNA mixtures, where the DNA of more than one person is present in the sample, are inherently more difficult to interpret than single source DNA samples. Often the way I like to think about this is in terms of comparing this to math. So we do DNA testing on single source samples. That's for like the purposes of databasing, putting reference samples into a database. This would be similar to doing basic arithmetic. We can say like 2 + 2 = 4. That would be like a single source DNA sample on that comparison. On the algebra side, we would be looking at, this would be similar to doing a sexual assault piece of evidence, where we have a 2-person mixture that might have high levels of DNA, and that would be coming from the victim and the perpetrator, the person that committed the crime, that would have those samples mixed together. And that might be algebra. And then the calculus would be like the touch evidence samples, where we have more than 2 people present, we have low levels of DNA, we have complex mixtures, sometimes or even individuals that are related to one another, and that complexity makes it more difficult to look at and to do. So when we often talk about DNA as being the gold standard, we're usually talking about that in terms of single source samples. That's the basic arithmetic. You need to have those skills and understanding to be able to do the algebra and the calculus, but realize that they're not the same, that there's differences across the types of samples that people are looking at. When we do the more complex mixtures, then we're getting down to or trace levels of evidence. We have higher uncertainty because we don't know how many contributors there are to the sample that's being done. This is just an illustration of the different parts of the process. So here on the left, we have the evidence sample. That DNA would then be collected and stored. It might be characterized, the sample, like if it's coming from a semen sample or a saliva sample. There might be the extraction and quantitation of the DNA that's done, the amplification that's next performed, then the separation part of the process. All of this is the measurement part of the process. So here's what we're gathering the data, we're generating the data to be able to look at. Then finally, we have the interpretation part. The second part of that is where we're taking the data, we're trying to understand the results, we do statistics on that data, and then we generate a report. If there's a person of interest that is being compared, then that profile would be compared to the profile from that evidence, and then a report would be issued, and that would be a part of a written report. So at the end of the process for a measurement, we have an electropharogram. Sometimes we label that EPG. This is the result that comes from doing the DNA testing. The second part of that, the interpretation, the end result is the written report. And that the things that are happening there is people are making decisions about what data to include, what kind of comparisons are being done, what statistical approach to use, and then how to describe those results to someone. Next, we have the fact that these things differ from one another. So we have, we're generating a DNA profile, involves measuring the properties of that sample. Interpreting the profile means that you're assigning values that are not inherent or not part of that sample. Okay, so that's an important thing to realize, that measuring a sample, generating that electropharogram is different than the part of the process where you're actually doing the interpretation. When the interpretation is done, the DNA analyst uses their judgment, they're using their training, they're using tools, including computer software they might have, they're using their experience, and they're considering factors such as the case context when they're doing this work. So in our draft report, we have some of the measurement factors that are being done, like peak position or the peak shape that you have. On the interpretation, there are steps that are involved with looking at, do you have a mixture? How do you call the peaks that are there? How do you infer the number of contributors? How do you estimate the ratio of contributors? Do you consider the combination of the genotypes that are present? And then comparing those to the reference sample that you're the person of interest, and then performing a statistical analysis of that comparison. Again, different parts of the process that are important, but things to keep in mind as you're looking at this. Again, you can go back and see this in the report, but there's factors that contribute to the increased complexity, and we have a key takeaway around this, that the process of generating a DNA profile can produce stochastic or random variation, and there's artifacts that can result that contribute to the challenge of DNA mixture interpretation. So these artifacts are highlighted up above. We have stochastic variation, we have stutter products, and we have sharing of common alleles. Those are some of the key factors that then influence, and we'll talk about each of these as we get more into describing mixture interpretation. There are, again, multiple factors that influence these things, as we've talked about, and this is part of what key takeaway 2.4 is around, that DNA mixtures, they vary in their complexity, and the more complex the sample, the greater the uncertainty surrounding the interpretation. So factors that contribute to the complexity include the number of contributors. If you're a two-person mixture versus a four-person mixture, there's a lot more complexity with that four-person mixture. The quantity of DNA that's being tested for each contributor, the ratio of those mixtures, of the contributor and mixture ratios, the sample quality, if it's a sample that's degraded because it's been out in the environment for a long time, and finally the degree of allele sharing, So how much do those contributors line up and share alleles with one another? So if you look at this in terms of the basic steps that you're taking from the sample viewpoint, on the top we have the Q sample or the question sample. On the bottom we have the listing of the reference sample or the known sample. And you go through this process for both types of samples before you do the comparison. You have to make a decision. Is this a peak or is it noise? Is this an allele? Is it an artifact? Is this a genotype? And you're making decisions about pairing the alleles, and that's what the computer programs are doing. And then finally, it can combine particular genotypes together across the loci to give you the profile, and then using the match probability to make some kind of assessment of the weight of evidence, and finally you write a report. So those are each parts of the process for doing this. And what's a key part here is that it's the genotype that matters, not the alleles. So how those are paired with one another, that's why it's called probabilistic genotyping, not probabilistic allele typing, right? So you're looking at the pairing of the alleles to figure out what contributors they potentially could come from. So it's the potential genotypes, not the alleles, that matter in a mixture. And this is why you're looking at those combinations of possibilities. So let's talk a little bit about this paper. It's freely available on the ISFG website. I put the link there at the top. And I would highly encourage, if you're doing mixture interpretation, you should know this paper. It covers all the main elements of what's being done. Again, published now 18 years ago, but it has a lot of useful stuff that's being talked about in terms of recommendations in terms of mixture. So I'll walk through, there's 9 recommendations. I'll briefly talk about them. These are the 9 recommendations that are part of this. And I simplified them just to illustrate the main points. But number 1 is that the likelihood ratio is the preferred statistical method for mixtures over what's sometimes called the CPI, or combined probability of inclusion, or what's sometimes called the random man not excluded. And the reason, again, is because you can use more of the data in terms of making that interpretation. And that's what the likelihood ratio enables you to do, is you're comparing the probability of the evidence given certain propositions or hypotheses. In order to use the likelihood ratio, the second recommendation is scientists have to be trained in and use the likelihood ratio. And then number 3 provides methods to assign the likelihood ratios of mixtures. So these things are cited, some references, and then again, this is 18 years old. A lot has happened since then in terms of the software that's been developed, but they still all use these same principles for doing that. Number 4 is that we follow the Clayton et al. Guidelines, and that's where and I'll explain those in just a second, to deduce the component genotypes. This is what's being done by the software programs when they're being used in this context. Number 5 talks about the propositions. If you're going to do likelihood ratios, you have to consider the hypotheses or the propositions. Sometimes this is called the proposition HP, which is the hypothesis of the prosecution, or sometimes it's called H1, the first hypothesis. And then the prosecution can determine its hypothesis or proposition, and the defense determines its proposition or hypothesis, and multiple propositions can be evaluated. So again, you can check and see if you want to consider a proposition where related people are involved, or a particular population is involved in your things. You can do that by doing the likelihood ratio with those inputs, those propositions. So the first 5 recommendations are really focused around the basics of using likelihood ratios. The second set are all around the data. So 6, 7, 8, and 9 are surrounded more around the data. And so we have, like, number 6 is when minor alleles are the same size as stutters of the major alleles, then they're indistinguishable. So you have to consider that a small peak may be a stutter, it may be a minor allele, depending on the ratio of the mixtures of the components that are part of that mixture. This becomes a key aspect of what's done with computer modeling for the probabilistic genotyping software programs. Number 7 is that we can only use allele dropout to explain the evidence if you have low signal data. So if you have a sample that's at several thousand RFUs, relative fluorescent units, you can only explain you can't use that to explain dropout for something else. You have to have a signal that's low, and that's probabilistically you have to consider it that way. And that's what the computer programs do. And the level, the height of the peaks that are present influence the probabilities that are being done of the potential combinations of genotypes. Number 8 is that no statistical interpretation should be performed on alleles below threshold. And what this means is that if it's not truly an allele, you shouldn't consider it. And the way you determine if it's an allele or not in the standard thinking is that you have an analytical threshold that says anything above that is something I can consider a peak. Anything below that is noise, is something I can't work with. And then number 9 is that stochastic effects limit the usefulness of the heterozygote balance and the mixture proportion estimates when you have low-level DNA. And again, this is why we use probabilities, because we can't be fully confident in the peak height balances because we have generally stochastic effects or the random variation in the PCR process. So again, these 9 recommendations provide the basic underpinning or the foundation for how mixture interpretation is done. And these things have been applied into computer programs, and that's what people have been doing in the last few years to use these. But you have to understand the likelihood ratio itself to be able to know how to work with this. So let's talk briefly about the Clayton guidelines then. This comes from the Clayton rules, or the Clayton steps in interpretation, come from a paper written by Tim Clayton and colleagues that were at the Forensic Science Service in England. And they wrote a paper called Analysis and Interpretation of Mixed Forensic Stains Using DNA STR Profiling. And these are the steps that they detail. And of course, the last one would be adding step number 7, which I showed you earlier, was adding the weight of evidence, or adding the statistical, once you've done a comparison, what's the meaning of that comparison. So we have to identify the presence of a mixture, identify and designate the allele peaks that are above of the threshold, that's detection of threshold. We then have to identify and estimate the number of potential contributors. That's important for calculating how many possible genotype combinations to be looking for. That often you do a relative ratio of the individuals contributing to that mixture, so you can see how is that performing across the different loci that are being looked at. Considering all the possible genotype combinations, depending on the context that's there, and again, based on the peak height ratios and so on. And then finally, comparing the relative of the reference samples, the person of interest, to that. Okay, so if we look at this in terms of comparing the expected and simulated results, if we're looking for how do we estimate the number of contributors? If you have 2, 3, or 4 alleles that you're observing at every marker, every locus across the profile, then it's likely you're going to have two people present. If you have 5 or 6 alleles at any locus, then 3 contributors are possible. There could be more if there's allele sharing. If you have more than 6 alleles at a single locus, then it's likely going to be more than 3 contributors. Okay, so there's been lots of simulation studies that have been done over the years. This is one I just selected to illustrate this from John Buckleton from 2007. And he found that a simulation of 4 people mixtures would only have a 0.02% would show 4 alleles or less. And that 76% would show 6 or fewer alleles for that. And so as you look, and it gets, you have to look at the overall perspective of this, but let me just illustrate on the next slide here with one examples from this paper. So this is what you see here is each row is a different marker. So you see D3 is a marker on chromosome 3, BWA, D16, and D2. And then what's being shown across the rest of that, as you go across the slide, you have the number of alleles that are detected would be 1, 2, 3, or 4. And if you have a simulated 2-person mixture, then we could say that around 1% of the time, the 0.011, that is, you only see, for D3, we'd only see 1 allele at that case, where we'd see 2 alleles would be 24% of the time, or 0.240. And then if we have 3 alleles, then we'd see those around 56% of the time. And then if we saw 4 alleles going across, then about 19% of the time. What you'll notice here is that the D2 marker and the D3, I'm comparing those with the boxes. The red boxes show that you detect 3 alleles at a locus around 56% of the time for D3 and only 46% of the time for D2. The 4 alleles, you're going to see 19% of the time at D3, and D2, because it's a marker that has more variability across the alleles that are present, around 44% of the time, you're going to see 4 alleles in these simulations when you have a two-person mixture. What that means is that a marker like D2 is likely to see more alleles when there's a two-person mixture or a three-person or four-person because there's more allele possibilities there. So if you were to look at this as a two-person mixture, we have what's illustrated there is D3 is there on the far left on the bottom, and 3 peaks are more common for D3. So in this case, we see alleles 15, 16, and 17. If you go over to D2, which is on the far right, we see that four alleles are more common for D2. We normally would see 4 alleles 44% of the time versus 19% of the time for D3. So what I'm illustrating here is that you see more alleles when you have a marker that has more possible alleles, and is more what they call has a higher heterozygosity. Okay, so when you're doing a mixture interpretation, this is an illustration from one of my books, is you have, if you were to start with a marker like D18, it's illustrated here, so we see 3 alleles, we see a 13, a 14, and a 17. So this is what we observe when we look at the electropherogram. What we want to infer then, the goal of interpretation is from that available data, we want to infer the possible genotypes and then determine the sample components. What went into that mixture in order to generate a result that has a 13, a 14, and a 17 at those particular ratios. So a couple things to keep in mind. We have the true sample components that are shown in blue up on the upper left. We have the sample processing process there. And then finally, we have the data obtained. So what you're able to measure is the part at the end, which is the data obtained. You have to go through this processing, and this is where validation, the validation studies that you've done, they help show the variation and the limits, hopefully, you've illustrated those limits of the process involved. So we have, in the true components, we have a potential of maybe a 12, a 13, a 14, and so on up to a 19. So those things that could exist. In this case, we would have a 13, 17 would certainly be a possibility. So we link those together, say this as 13, 17 genotype. And then if we have a 13, 14, those could go together. And then we have a mixture ratio between those. So in this case, we have allele sharing and overlap or stacking. That's where the 13s from one component line up with the 13 from the other component. And that then leads to a slightly higher 13 in this case. That will depend on both the number of contributors and the mixture ratio that you're looking at and the sample components that are part of that. So we also might be able to say that the larger component in this is a female because of amylogenin testing that's been done. And then the smaller component might be a male. In this case, again, for the amylogenin testing that's been done. In this case, the ratio might be four times the female to 1 time of the male in terms of the amount, the relative amounts of sample. But what matters here is that we're going through a processing of copying that DNA. And this is where the variation can occur. So we can extract how much DNA gets extracted from sample to sample can vary. How the PCR process works can vary. So we have an overall total amount of DNA that we're going to copy. But we have also the injection part of the process, and then we have the detection part of the process. And this is where validation studies have illustrated and shown something around these pieces, but that's what then influences the final result that you get in the end. So again, we're trying to backtrack and figure out what's on the left side of that slide, but we have to rely on what's on the right side of the slide to make those decisions. So here's just an illustration then with allele sharing and a 4-person mixture to show some of the complexities that can exist. So if we have one marker, D16S539, and we have four-person mixture, we have contributor ratios of 2 to 1 to 1 to 1. These are the genotypes that are being illustrated here at 12, 13. You can see there's 13s that overlap with each other. So if we were to look at this, we would, if we had fully proportional signal between the contributors and the individual alleles, we might see the 10 and the 13 here are shown in red. The dark blue is the 11, 12. The green is the 11, 13. And then we have the 12, 13, which is the two times that amount, because it's a 2 to 1 to 1 to 1 mixture. And this is where we expect to see something along these lines. And then if we were to look what's here in the middle, we have the expected quantitative signal due to allele sharing. And this is what we'd expect to see in the signal if everything worked as performed. However, if you were to actually look at what happens here, real data will show variation. And that's because of stochastic effects or random effects that occur during the PCR process. So here, what's illustrated here is the alleles 10, 11, 12, 13, and you don't see this what you would normally have expected to see here, as the 10 is the smallest, then the 11, then the 12, then the 13. But in fact, you have the 11 and 12 being almost equal in height. The 13 is lower than what you'd expect. And this is what's happening in the process of generating that data from that mixture, this four-person mixture, as part of this process. So we see the complexity that exists in these samples, and this is what makes it challenging, and certainly to do this by hand. This is why probabilistic genotyping is applied, but then what types of validation have been done to make sure it works properly. What this slide is to illustrate, the detectability of alleles can vary based on the amount of DNA that you have. So if you have, this is on top, you have 62 picograms or 0.0625 nanograms. Then you have, below that is 125 picograms or 0.125 nanograms. Then 250 picograms, 500 picograms. And what you'll see from looking at this is that there is variation in the amount of DNA that you're seeing. In terms of the peak heights are changing, you're seeing more peaks. By the time you get down to the 4 nanogram level, you're detecting a lot more peaks from this. Whereas in the original one up here, you don't detect them out as much. So the amount of DNA that goes into the testing has an influence. It will also impact the peak height ratios that are being seen. And that then influences the variability that will exist in those samples. There's been a couple other ISFG DNA Commission articles on DNA interpretation, mixture interpretation. I just wanted to point those out. So I encourage you to read these to study more about dropout and drop-in issues. And this paper in 2012 talks about that, where he talks about probabilistic methods can be used to weight these things. We have dropout and drop-in probabilities that have to be based on validation studies. So what types of studies have been done to look at that? We have the weight of evidence should be expressed using the likelihood ratio principles that we mentioned. And then finally, use of appropriate software is highly recommended so you don't have to do this by hand. Then we have a software validation, and recommendation 12 points out that the laboratory should consider the range of samples expected to be analyzed in casework so that you can define the scope of the application of the software. And then we want to make sure we address true donors and non-donors, and also related and unrelated individuals, and cover a range of possibilities that you expect to see in your laboratory. Then more recently, in 2018, there was another DNA commission that talked about the difference between investigative and evaluative reporting, and then some of the common pitfalls that can occur when we were formulating the propositions for this, and also discussing the impact of low-level mixtures and the challenges there. Then in 2020, there was an article that was published on activity-level propositions and distinguishing between the results, the propositions, and the explanations. So if you're going to get into mixture interpretation, you really should know these articles, because they form the basis for what's being done in your work. All right, so now I want to change gears a little bit and talk about the question you're trying to answer. What is the question? So this depends on the inputs. Likelihood ratios always depend on the inputs and the questions that you're considering. So what question we're answering depends on the propositions or the hypotheses that are being made. So you need to consider the hierarchy of propositions that were described back in 1998, which come down to source, activity, or offense. And the likelihood ratio is the probability of getting the evidence if the defendant, for example, if the defendant is a contributor, as compared to the probability of getting the evidence if that defendant is not a contributor. Again, this involves assumptions as to the number of contributors in the mixture, and it's influenced by a number of inputs. So again, to point out, there's a book that was published by Joe Bright and Mike Coble a couple of years ago that talks about this, and they use the word assigning, not calculating. And they point out that these propositions are set based on the relevant case information. That we normally would know the prosecution's case. We may not know the defense propositions. And so you can propose something, and this would make sense from the most sensible proposition, but you don't know if that's necessarily what the defense will choose. And this is part of the challenge that exists with using likelihood ratios. What this slide is to illustrate is all the different components that go into a probabilistic genotyping DNA mixture interpretation. So up in the upper left-hand side, you have the mixture occurs, where you have cells from multiple contributor co-deposited. We have the sample being collected and recovered on a swab. We'd have then the data being obtained, and this is then generating the electropherogram. Then a decision has to be made of the level of input data that's determined by the lab. That's where, defined by the validation studies, you make some decision about the threshold of data that goes into this. Then what's in a box here is your probabilistic genotyping software. This is defined by the validation studies. And you have model parameters like what peak heights or what stutter percentages that you'd be using, what's the probability of dropout. These are what are called biological models. Then you have the weighting of the genotypes that are used by the computer program. This is only done once you've established the gray box up here above this is the number of contributors, and you have to make some kind of estimate. So make an assumption based on the electropherogram data to say this is a 3-person mixture, or I'll test this as a 4-person mixture. Then we're able to generate the weighted genotype possibilities. The propositions are then set. They have to be decided that, again, this is based on the number of contributors and the case-specific situation. You then have inputs in terms of your allele frequencies and the reference profile that you would be looking at. And then finally, a likelihood ratio is assigned with these statistical models. A report is generated. Sometimes verbal equivalents are added to that. A testimony can be offered if it goes to court. And finally, the trier of fact has a decision to make considering the DNA information along with other information. So the point of this figure is to illustrate there's lots of pieces of information going on. And what's in gray is the input that you have. You have the input of the data, selecting which peaks you're going to look at. You have the input of the number of contributors. You have the input of the propositions. You have the input of the allele frequencies, and you have the input of the reference profile. All of those influence what that likelihood ratio assignment will be when you're doing a mixture interpretation. So that's why you have so much variation in terms that can be applied in terms of, and then at the end of the day, though, the trier of fact has to make a decision about their work. And I just illustrate this with, and I think I'll just finish with, after I do this, I'll do an example and I'll stop there, and then you can take questions if you want. I have more material, but I think that's probably enough for today. It's probably challenging with the interpretation of the language as well. We have, I just put in here what you start with. You have the data, the modeling, and then the allele frequencies. And then you have the data you obtain from that. You have the reference profiles. You have the number of contributors that are the decision made there. And then you have the propositions. Okay, and finally, the weighted information. I'm going to illustrate that with an example on the next slide that goes to these points. So what the point of this is to illustrate what questions you're answering will vary the results. So the evidence is obtained. That's like the little one going back to the previous slide. And here's an illustration with 2 loci. So we have D16 and D2, and this is what the electropherogram would look like for those. And this is from chapter 7 of this book. I just illustrated this. All the peaks are above the threshold. So our threshold is set at 50 RFUs. So all these peaks are detected. We're going to use these allele frequencies from the FBI Caucasian database. Again, that's a decision that was already made beforehand. That's why it's listed as a 0 in green, because the lab made those decisions. We're going to use this analytical threshold. We're going to use these frequencies. The person of interest then is someone that has an 1114 and a 2123. So they're both present in the sample. Those alleles are present and the genotypes could be present. Again, that's a decision from you have a reference sample to compare to. Now, if we assume 2 contributors, then again, that's an assumption that's being made. Then we set the propositions, and we also set the theta correction, which is the subpopulation correction. And then we determine the genotype weights, which would be estimated by a process of generating these weights in the computer program. Okay, so if we generate these, the possibility of an 1114 and a 1213 being present in a 2-person mixture with that particular combination of evidence would be about 68% of the time in terms of the simulations that were done inside this computer. The presence of a 1314 and an 1112 genotype would be about 15% of the time, and so on. And you can see down below the D2, we have a 2326 and a 2121 being the most common, but the 2123 and the 2126 is the actual, because I know what was put into this, so you can kind of say, here's what it would actually be, where the arrows are. Then if we were to calculate a likelihood ratio under these assumptions with this theta correction, we get a likelihood ratio of 1,144. If we calculate this with just what's called the product rule, where we have the theta correction set to 0, our likelihood ratio is higher, 2,895. If we condition on the victim, so we have more, we reduce the uncertainty in the results because we condition on the victim, and the victim in this case would be a 1213 and a 2126 of these 2 markers. So therefore, we can that sets aside that information. It gives us more power. So now our likelihood ratio goes up to 4,143. However, if you consider a possible untested brother as one of the hypotheses where you're dealing with someone that might be related there, now your likelihood ratio goes down to 8, just 8. And so that illustrates that the decisions you're making in terms of the propositions, what's being set there on the right side of the screen, have a huge influence on the final result. So you have to think about each of the components that's going into generating that likelihood ratio, and are those appropriate to use in that particular case? Maybe they are, and maybe they're not. Depends on that particular situation. So this is, again, all the same data, but you're using different assumptions and different propositions in making doing this work. So these are all values that reflect the subsource likelihood ratio. This is not an activity level. They're looking at from the evidence from the standpoint of, could this particular scenario be, did someone handle this bike, or just was it transferred, the DNA transfer? What are the propositions that went into that for an activity level? Those are very different. All you're saying here is that sample, does that DNA, is it present? Not how did it get there, or when did it get there? You're not answering any of those questions. You're just answering, could that particular person's DNA be present under these assumptions, under these propositions? That's what you're answering. So this illustrates there's a lot of components that go into, and you need to understand these when you're doing the calculations that go into generating that result. You can see the variation can be several orders of magnitude, from an 8 to 4,000 on just 2 markers with this. And of course, it will get wider if you do more and more sections of DNA that are being tested. I'm running out of time, but I want to just finish with this, that there's the level of hierarchy of propositions. The questions we're answering mostly are subsource questions. Is the DNA from the person of interest? That's the question you're answering. You're doing that with the DNA profile. And this occurrence of DNA profile in relevant populations is what you have to look for. If you're doing source, you have to know something about the biological fluid that's coming from. So is there enough DNA that you can definitely associate that with semen or saliva? And so, is there any cross-reactivity that may exist? If you're trying to say, did that person of interest perform the given activity, then that's an activity level question, and that requires information not only on the source factors, but over there on the right, you see you need to have DNA transfer, persistence, recovery information. You need to have DNA present for unknown reasons, like the background DNA. And then again, this is all something coming from that DNA interpretation or the DNA commission article. So this is all we're usually answering when we do tests is we're answering, is the DNA from that person of interest? We're not answering, is it from a particular biological fluid or did that person perform an activity? That requires separate questions or separate sets of data to be able to answer that question. So we have an impact of DNA transfer on those things. And again, we can also do sub-subsource, which is done when we only look at a portion of the mixture, which is often done with software today. So hopefully this is helpful. I'll stop here because I know we're out of time. There's a number of other slides, but I wanted to I'll just drop down to the end and point out this here at the end that we have a sense about science has published this article with, and again, this is the one that's also in Spanish, but the final point they make is as DNA profiling continues to grow more sensitive and is used in more investigations, the need for accurate communication between the scientists and the non-scientists only grows, both to ensure that the expectations of the technology are realistic and that its limits are properly understood. So if you're going to do DNA mixture interpretation, you want to understand its limits, and you want to understand what can you get out of that sample and what can't you get out of that sample. So with that, I'll just thank you for your attention, and again, happy to take questions, preguntas, for things. So thank you. I'll stop sharing here. But you have the slides. You can go and feel free to email me and ask me more questions if you want.
Muchísimas gracias, John. Yo creo que tenemos tiempo para una pregunta por parte de la audiencia. Bueno, yo sí tengo una pregunta. Ya. En cuestión de los software para interpretación de mezclas de DNA, existen dos opciones que es el RMX Studio y hay un modelo continuo que es conmigo. Aunque en la práctica he escuchado que la gente prefiere el RMX Studio, que es mucho más fácil de usar. La pregunta sería si realmente tú recomendarías hacer un análisis hacia un modelo continuo, en el caso de Euroconmigo.
So the questions around the software. What software is good to use?
Sí.
Yeah, I can't recommend any specific software. Again, the NIST disclaimer, I can certainly say there are programs available, and we have in our report, we talk about a number of ones that are available. I've written some review articles. There's probably 15 or 16 different programs that exist that could be used. Some are going to be tested more than others. So it depends on what your needs are and how much money you're willing to spend, or doing open source where there's freeware, open source software you can download and then use, but it doesn't come with as much support. So one of the things, if you buy the software, the company will typically give you more support, but it's going to cost you more.
Ok, muchísimas gracias. Y bueno, si no hay alguna otra pregunta, estoy buscando aquí en el chat. No veo. Bueno, muchísimas gracias por tu presentación maravillosa, John. Espero verte pronto. Y también esperamos que en algún momento puedas venir a México, que estás cordialmente invitado. Vamos a hacer todo lo que ya por acá. Muchísimas gracias.
Okay. I got a question. Could you choose the option of interpretations, the button that looks like a world? Oh, for me, let me see here.
Okay.
Yeah. Okay. I think I have it now. English. All right. Yes, I can.
No, tengo que repetir otra vez porque no escucho, ¿verdad?